appreciate very much this environment. It's fantastic to be with all the people and so I'd like to talk about cell-based modeling of wound contraction. So I have a project in the Netherlands from the Dutch Burns Foundation, and I'm interested in burns, so in wounds, injuries from burns. And I'm also interested in angiogenesis. I'm also interested in cancer. Okay, well, of course, cancer is something that affects a lot of people, so it's a very important disease. Okay, so let me start with a small cartoon. So, does it work? Yes, it does. So, Amit and I have been collaborating for 6 years, and we developed several models, and we developed continuum scale models as well as cell-based models. And this is one of the cell-based models, which kind of models the initial stages of cancer. So what you see here are the green cells. The green cells are, let's say, lung cells. They could be epithelial cells. And the red cells that you see here are cancer cells. And here there are some points where the blue cells are released. You might have seen them. And these blue cells are T lymphocytes. They represent T lymphocytes that actually try to find their way to chase these red cells, the cancer cells, and try to eradicate them in such a way that cancer can be battled, right? So it can be fought. Well, of course, this model is a very simplistic representation of the disease. This was actually constructed by one of my bachelor students, so a third-year mathematics student. But it represents, it illustrates very well, very nicely what it actually is. And what we see is that this type of modeling, this type of cell-based modeling, can also be used, for instance, in modeling closure of gaps. You can think of wound closure, for instance, the closure of epithelial wounds. We also apply this to the contraction of wounds, wound contraction. So I'm going to show that a bit later on as well. But I was actually always kind of inquisitive, curious, whether or not this type of modeling could be applied to a process like angiogenesis. And angiogenesis is, well, I'll show you. Angiogenesis is actually nothing else than, so imagine that you have this kind of blood vessel, this kind of set of blood vessels, and then here you actually see that these blood vessels are sprouting. So you get sprouting of new blood vessels from pre-existing vessels. This is actually angiogenesis. And this is a process that takes place all over your body. And in some cases we are very happy that it takes place. It's very important because we need a good supply of blood everywhere in the body. But in some cases, and I will show that later on as well, in some cases you don't actually want this to happen. For instance, in the cases of tumors, cancer. So what you see here, basically, this is a very schematic cartoon of how angiogenesis can take place. So there's a triggering signal. You can think of a chemical, for instance, or a mechanical signal. And this signal is kind of received by the endothelial cells, which form the backbone of the blood vessels. And then these endothelial cells, they start dividing like crazy. They start getting mobile. And what happens is that they kind of form new networks. And that's actually what you see here. That's actually the end result. But this is I was actually wondering whether or not our cell-based models were capable, could be used in order to model the process of angiogenesis. This was actually one of the research questions that I had. I am not going to say yes yet, or no. Okay. So let me sketch the process of angiogenesis as a sub-part, as a sub-process in many processes in the human body, in mammalian bodies. For instance, you can see here, this is a schematic of a tumor. So what typically happens is that if you have a tumor, then in the early stages, it would start, oh my goodness.
No.
It would start, that's normal, okay. So if you look at let's say the volume of a tumor, as a function of time, in the early stages, you will see that the cells are dividing and then, well, the tumor is actually growing. It's kind of expanding, it's growing. And then, at a certain moment, the cells will need more supply of blood. And in that case, the tumor cells no longer divide, and even some of them will die. So what you get is that So here actually, you would think that, well, okay, nothing happens anymore, the tumor is not dangerous. However, in the course of time, the blood vessels will arrive at the tumor, and what happens is that, of course, by the arrival of the blood vessels, the tumor cells will be supplied with oxygen, with nutrients, and they will become very happy. And when they are happy, they start dividing like crazy. And then they start getting mobilized, they start dividing, so what happens is that the volume goes up again. And this is actually the moment on which a tumor can become dangerous. So it is also able to spread out through other parts of the body. And this is the cartoon actually that I wanted to sketch here. And then the tumor becomes dangerous. Well, this is a case in which angiogenesis is an undesired process. So here you would like to inhibit angiogenesis if possible. And there's also medicines, medications to try to inhibit breast cancer, for instance. But in some cases, these medications didn't work well. Another thing where it happens is, for instance, if you have an injury. Imagine that you have a wound. Imagine that you have some injury. What happens is that, well, there's a lot of chemical signaling, there's mechanical signaling and so on. I'm not going to talk about it right now, but there's a lot of processes going on. And one of the processes is the restoration of this vascular network. So here angiogenesis is very important. It's a very important process. And so it's a generation of new blood vessels for the supply of these nutrients and oxygen for the wound healing. So it becomes very important here. So I'm going to talk about angiogenesis as well as wound contraction. So I'm actually giving 2 talks at the same time now, but I've got an hour, I think, right? So there will be wound contraction. And what is actually wound contraction? Well, wound contraction actually happens if you have a serious wound, if you have a big wound. You can think of a big injury. And what physiologically actually happens is that the skin will contract. So the fibroblasts, which are, let's say, the main cells in your skin, will pull on the extracellular tissue in such a way that actually, for instance, if I would have had a burn here, I wouldn't be able to lift my head anymore. And this is a very serious problem. So I have seen some patients in hospitals, because we are collaborating with medical doctors, with plastic surgeons, and it's really bad what you see over there. So people are really losing their mobility. As well as, of course, that burn wounds are very ugly. So you are left with hypertrophic scars, which make the face of a patient very, very ugly. So it's also an aesthetic component. And we are talking about wound contraction if it as a process during wound healing which leads to skin constriction. The doctors refer to a contracture, a wound contracture, so not a contraction, but a contracture, if the wound contraction leads to functional limitations. So then you have a serious problem, right? And you mechanically have a serious problem. Well, so what we need is actually, well, in our model, we try to incorporate things like mobility of cells. We try to incorporate divisions of cells, death of cells, but also differentiation of cells. So one of the projects that one of my MSc students carried out was a project on the construction of a model for cell differentiation. And actually we were also looking at the application that Ami just showed about fat, about adipocytes and muscle cells. So I will show some simulations later on this topic. Maybe it's three talks in one. So what you can have is, for instance, you could start with the mesenchymal stem cells, which are capable of differentiating to fibroblasts, or to adipocytes, to chondrocytes, and to all kinds of cells, to muscle cells, etc., etc., etc. So we try to develop a formalism for this. So I might say a bit more about that later on. But the main part of the talk is angiogenesis, like I mentioned earlier. So we collaborate with the group of Sue Gibbs. She's English, by the way. And she works in Amsterdam at the Free University. And she's an expert on skin. She's an expert on experiments on skin, both on large-scale experiments as well as on small-scale experiments. And she's a very, very clever lady. And what we are considering here is, so I sent one of my master students to her lab and to do some simulations to have a good look at what is going around there. So it was a mathematics student who was also looking at the microscope sometimes, because I think they should do that, right? Mathematicians, you should also have a look at the microscope. I also like to do that. So they look at the microscope, and he also tried to figure out what was going on. So what they did actually was the following. So they did a lot of experiments. So here you can see 12 times, let's say, 8 assays in which they put some fibrin gel. So what they did, they had this circular assay, this cylindrical assay where they put fibrin gel in it, and on top of it they poured some extracellular liquid. They call it extracellular liquid. It's a watery substance, and this liquid contains a solution of endothelial cells. And then what they do is they let these endothelial cells just move down towards the top of the fibrin, and then they look what they are going to do. And so this is actually, so Sue Gibbs refers to this as modeling. This is a model. This is a model, okay? This is a model. It's not a mathematical model, but it's a model.
Biological model.
It's a biological model. Yeah, it's a biological model. And, well, this is actually her PhD student, Hanneke Monsieur, who actually carried out the experiments, because Sue doesn't do them themselves, of course. And she also added tumor necrosis factor and vascular endothelial growth factor, or either of them. And the idea was to stimulate the endothelial cells in such a way that they will form networks. So that's actually the idea that they have. So they analyze this experimentally, and what they analyze is, so they use microscopy, and so here you can see a micrograph of what they analyze, and this micrograph is a micrograph of the top layer of the fibrin domain. And so this fibrin domain, so the top layer that they analyze this, and here you can see, if you have good eyes, you can see the endothelial cells. And this is just a magnification. This is a magnification of this thing. But now, this is the initial situation where the endothelial cells have not been stimulated yet with these growth factors, with vascular endothelial growth factor or with tumor necrosis factor. But after a couple of days and after stimulation with vascular endothelial growth factor or with the tumor necrosis factor, then you can magnify this, and what you see here are cavities. So now these cells have kind of drilled themselves into this fibrin medium, and they form channels through this, and these merely are the blood vessels. These merely represent the blood vessels. And this is something that we wanted to model. Okay? So I sent my student to this free university in Amsterdam, the medical center, and we talked about how to model this. And so we constructed a model for this. Okay, so let me describe the model a little, and well, be patient because I'll come up with some equations. Don't worry. And so first, I'll just show you some cartoons, and later on you will see some equations. Don't worry. So the model is actually as follows. So we have all these kind of small-scale biological processes that are occurring in angiogenesis. So what we have is proliferation of endothelial cells, cell migration of tip cells and stalk cells. It turns out that if you have a layer of endothelial cells, that some of these cells, just like people, like Amit also said, these cells are like people. So some of these cells will take a lead. They take the initiative, whereas the other cells will just follow. So, and these are the same cells, but they act differently. And what is actually going on there on a cellular level, I don't know, but I only know that they are doing, that they are behaving like this. That's the only thing I know. That's the only thing that they know as well. And so what happens is that these tip cells and stalk cells, they will migrate. It's like chemotaxis. I don't know. I think all of you know what. I think everybody knows what chemotaxis is, right? So they will migrate according to concentration gradients. There will be durotaxis. They like to be stuck in a nice rigid atmosphere, so they will move towards higher stiffness, not too high, but I think I'll say something more about it later on. But they also show random walk. Okay, so there's also random walk, and so they walk like drunk people. So they are really human, I think, in a certain sense. They also exert cell forces. So I'll also say something about that. And this is actually a cartoon. The tip cell will kind of chemically dig a hole here, will move into the fiber network, and then the stalk cells, they will follow. That's actually the picture. This is actually the picture. If you understand this, then you actually understand the modeling that I'm talking about. Well, there's a lot of models in literature, a lot of models. There's a whole jungle of models concerning angiogenesis. So you can, for instance, think of the cellular POTS models, which have been developed by Glaser and Grenier in 1992. They applied these models for the first time to tissues, as far as I know. My friend Roland Merks, you can see him here, there's Roland Merks, he's working in Amsterdam, and from Kolwijk. They developed some models for angiogenesis, as well as Rens, who was one of my previous master students. He's working with Roland at the moment. She also was involved in the development of cellular POTS models. So these cellular POTS models, I think you know them, right? So these cellular POTS models are models which consider a lattice, and each lattice point is either occupied by a cell, for instance, or not occupied. So it's kind of a digital model in that sense. There's also continuum-based models. So you can think of stochastic principles here, but also some deterministic principles like energy functionals, and that's actually what Merckx is doing. There's also continuous models, which are entirely based on partial differential equations. You can think of the work here in Britain also by Gaffney, by Patet. You can think of the work by Makelakis, and you can think of many works. Here, the list that I'm mentioning here is, of course, far from complete. Far from complete. And what I want to do now is I want to use a semi-stochastic model, which is also semi-continuous in the sense that we consider cells as spheres. So I'm going to consider to treat cells as just ideal spheres, okay? Ideal spheres that are able to move. So it's a particle-based model. And so that's actually what I want to do with this angiogenesis process. Yeah, my student also carried out some, so we tried a bit, you know, to see how, in the framework of a literature study, I wanted him to work on several models. For instance, here, this is an example of a cellular automata model, a cellular POTS model, which is lattice-based, where we have some cells on top. So this is the fibrin, the extracellular liquid, and here the cells are digging a hole, and we can actually model that very nicely. This is actually the model by Roland Merks, which was programmed by my student, by Frans. I will show his picture as well because he deserves that. So what we do is we distinguish between tip cells, stalk cells. We have the substrate components as well, where we distinguish between the fibrin, the boundary membrane, which is on top of it. This is a thin membrane. I think we could also leave that out for the description, and the extracellular fluid, which is on top of it. And all cells, in this part of the talk at least, at this part of the talk, all cells are spherical, in this part of the talk. So later on I'm going to relax this as well, Well, the modeling issues are, yeah, I know people are getting impatient for the equations, but the modeling issues are, we have cell migration, we have differentiation of cells, we have traction of the cells on cellular matrix, plastic deformations, apoptosis, cell death, secretion of chemokines, as well as a mechanical balance. Okay, mechanical balance as well. And because these cells, well, I think Amit told you about it in his previous talk, exert forces. Everybody knows who this is, right? So we have this. He looks compressed, yeah. I think that was at a time that he didn't run that much, right? I think. And so you started running quite lately. I think this is one of the influences that I have on you, I guess. So we have this, these spherical cells, which are portrayed as circular here, which could also jump into one another. And then we also have some contact mechanics model that actually prevents them from colliding too much into each other. So we have these contact forces, which are dealt with by Hertz's contact mechanics model, by the use of the local cell curvature. And we also compute a field of a strain energy density by means of a finite element solution and gradient recovery in some cases, and in some cases we just use Green's functions. So we use different approaches.
What do you mean by the field strain energy?
So what I mean is that we compute displacements everywhere, and from that we can compute the strains and the stresses, and then you can compute the
in the membrane or the fibrin?
Yeah. Well, the mechanical forces by the cells. So this is done by, this is a PhD student of mine, one of my PhD students, Daniel Koppenol. He's a very brilliant guy, I think. He is actually working on the project of burns. In fact, he works on continuum scale models, but we borrow some of his formalisms also. We also extend or transfer some of his formalisms to cell-based modeling. So we have these mechanical forces by cells. So this is the force balance. So F is the represent all the body forces that we take into account. Well, with respect to burns, I'm not going to mention this here any further, but in burns we distinguish between two different types of forces. So there's one force, which is the temporary force, so cells that are actually pulling, and you can think of the fibroblasts, the myofibroblasts, who are pulling on the substrate as they are moving around in the wound, as well as plastic forces. You should realize that that I'm going to do something dangerous, so what happens is basically the following. So can you hold this for me? Oh, can you just, or maybe maybe not you. Yeah. Can you hold this? Okay. Okay. So what happens is actually the following. So now imagine that this is just a collagen, right? This is a long molecule, big molecule, and now what happens is
you unplug everything.
Yeah, maybe. Yeah, I think so, right? No. No. What happens is the myofibroblasts, they are doing the following. They are actually shortening the chains. Okay, and you see, indeed, our colleague is holding it, but they're shortening the chain. So you get these. So indeed, maybe it's better to take it back. But so what happens is that indeed these chains will be shortened, and there will be. And all the tissue will be left with some residual strains. And this is exactly what happens. And this is why you have these strains in your body and in your skin, and you're no longer able to lift your head. And this is actually a big problem. Okay, so this is one way to model it. And of course, you could also use morphoelasticity. This is actually what we are doing at this moment in Delft. And so we use a more advanced modeling framework as well. But this is actually what we did in the first calculations. So this is this plastical term. As well as we have some boundary conditions, spring condition, and we have these temporary forces like I mentioned earlier, and these plastic forces. In this talk I will limit myself to the temporary forces only. Okay, so if you want later on I can also say something about the plastical forces as well. Okay. This is another student. This is my master student, Wietse Boon. Very nice guy, and what he helped me with actually the mechanical framework of all this, and so the mechanical forces. So he graduated a year ago, and at this moment he's in Norway. Okay, so he went to Norway. He's doing his PhD study there, and so I was there a couple of weeks ago as well. And he's having a very nice time. He's having lots of fun there. I also had lots of fun there. And so what we did was actually the following. So we developed a mechanical model for cell forces that are exerting their forces on the extracellular matrix. So the idea is actually the following. So now I think it's going to, yeah, this is normal, I know. So what we actually try to do is the following. So imagine that here we have a cell, okay? And you can think of a fibroblast, or you can think of a myofibroblast, or whatever cell you like. And this cell is exerting forces on the environment. Okay, you can think of pulling forces, for instance. The pulling forces that are exerted by the fibroblast in the framework of wound contraction. So what we did was the following. So we divide the cell boundary, and we also did this in 3 dimensions, into elements. Okay, so you can also think of this in a 3-dimensional representation. And on each of these elements, okay, so you can define the outward pointing normal vector. Okay, so you can define a normal vector. And on each of these elements, let's say, we let the cell exert a certain force. And so you can think of, for instance, a force that is exerting away from so in a negative normal vector. So let's say here we have a force. So let's say this is element I, okay, and this is cell J. Okay. So we have this Fi, which is in the direction of, let's say, the normal vector, unit normal vector. So let's say it's kind of a certain pressure, P, okay, on T and on Xi, okay, in the direction of the normal vector. So this could be negative. If it's negative, then of course it's a contractile force, okay, and so on and I, of course. And furthermore, okay, we also assume that this P is a force per unit area or unit length. So we also get, let's say, the distance or the area of these elements. And so let's say delta comma I, could be an area or it could be a length or whatever if you're in 2 dimensions. And furthermore, we also have this force balance. And so we have the force balance like this. Okay, so this is our force balance. And what we do next is, okay, so well, we assume that this is kind of a point source, x minus xi. So this is the Dirac delta function, which means it's 0 everywhere. So everywhere in this domain there is no force, except on this point.
Are these x bar, the x, are they internally in the cell or in the extracellular matrix?
You mean the x? They are just on the cellular surface,
Okay.
which is embedded within the extracellular matrix. So actually I should say xij. Okay, yeah, that's mathematicians here, of course. So they, I should tell them everything, of course. Okay, so then we get this. Okay, and then of course, what we do next? Well, this point is acting a force, but the neighboring point is acting a force as well, etc., etc., etc. So that means we add all these forces. And so we sum over all these forces. Okay, and we get the force exerted by cell J. Okay, so we get the force. And then, of course, if we have multiple cells, there's no problem. If we have more than one cells, if we have a multi-cell colony, then of course we have to sum over all these cells as well. So in fact, we have to sum. So then, of course, we are also going to sum here over all these cells. We do the same thing here over all these cells. And then we have our body force. Well, and you can also, this looks very much like the approach that is applied in fluid dynamics. So the immersed boundary approach from fluid dynamics. And what you can do, of course, you can also formalize this a little, and that's also what we did. So we let actually the delta comma I, we let it tend to 0, which means that the number of points or number of elements on the cell boundary tends to 0. And then, of course, you get a limit situation, and then you get just a Riemann integral. So in other words, and we replace this with an integral. So then you can replace this with an integral. Should come back, right? Yeah, and that's what we did here, right? So that's what we exactly did here.
So here you have no cell-cell contacts.
Sorry?
You have no cell-cell contact here? Or?
So yeah, in this framework, there's no cell-cell contact, indeed. However, of course, it might happen. You might have cell-cell contact, and there we use the contact forces, the contact dynamics. Yeah. Okay. But you know, you can do many different approaches for this. An alternative approach could be that I say, well, okay, so I have my domain of computation, and then I consider the cell as a whole, as a cavity or a hole, and then I use a boundary condition that describes the force that the cell is exerting. But then, of course, if the cells are moving, then this would be quite a hassle. Okay, this would be quite a hassle. So this is I think it's a very simple approximation, because I'm just a simple. Amit is a simple engineer, he said, but I am just a simple mathematician. Okay, so I'm a very simple guy in that sense. Okay, so yeah, so this was the mechanics. We also, of course, we also have the chemicals. So we have this vascular endothelial growth factor, which will attract the tip cells to move downward. Just look at the time. We have MMPs. Well, MMPs is a chemical, which actually is a protease. So Sue Gibbs is always correcting me on this. If I use the word chemical, she's not happy, but in fact it's just a chemical for me. It degrades the boundary membrane, okay, and this is secreted by the tip cells. So the tip cells, they secrete these MMPs, and these MMPs will actually degrade the boundary membrane, so this top membrane. There's UPAs. Don't ask me about the abbreviations. I can look them up for you, but these are big words, and I would almost break my tongue by pronouncing these names. UPA, which will degrade the fibrin here, and this is also secreted by the tip cells. And then there is the DLL4 or DLL notch, and this DLL4 will actually make the stem cell. This is also secreted by the tip cells. Will actually are responsible for the generation of a concentration gradient in such a way that the stalk cells will actually follow the tip cells, and so that the vascularization process can take place, is able to take place. Well, here I did not incorporate mechanics yet. Okay, this is just chemistry, but of course I'll say something more about how we incorporate, how we link chemistry to mechanics as well. And we use the framework of Anotida. So this is I'll show that. It's very nice, I think. So this is the concentration of the vascular endothelial growth factor, the DLL notch, and UPA. I wrote it down here, and also the MMP. So it's kind of diffusion equations, where we got values for the where we were able to acquire some values from literature for the diffusion coefficients of the different components for these chemicals. But of course, there's always a wide variety, so that's a big problem, in fact. There's a big variety, but we use some values that were documented somehow, and we used because we use kind of an hybrid model, in the sense that an alternative could be, so what you could do is you could actually treat the decay of the fiber network, you could actually treat it like a moving boundary problem. You could treat it like a moving boundary problem, and that's what people like in this community. You could treat it this way, but we didn't do that. So what we did, we actually tracked the volume percentages of all these phases, of the fibrin, of the boundary membrane, as well as of the extracellular fluid. And so we adapted the diffusion coefficients according to mixing rules. And we did the same thing with elasticities and this kind of thing. So I think this is well accepted. And then there's also reactions. So this is actually the consumption of the vascular endothelial growth factors by the tip cells, and there's by all the endothelial cells, and this is a secretion, and this is secretion by the tip cells, and here once again secretion by the tip cells, and also once again the uptake by the stalk cells here. And here you can see actually the loss of, for instance, UPA as a result of the degradation of the boundary membrane, and same for the fibrin. I will show the equations for the fibrin just now. And here you can see the equation for the fibrin, which is degraded by the UPA, as well as the boundary membrane degraded by the MMPs. And here, of course, once we lose MMPs, we lose boundary membrane, and then we assume that this space is occupied by the extracellular fluid. And of course, it's a loss there, but that's just on the top, so we are not considering that. Okay. Well, we also need to model the migration of the cells. So, we treat the cells as particles. So here we have a term which incorporates the mechanical forces as a result of contact between cells. So this is the contact model by Hertz. Here we have chemotaxis for the tip cells and for the stalk cells. So this is for the stalk cells and this is for the tip cells. As well as here we have Fs. Well, Fs is the volume fraction of the solids, by which I mean the boundary membrane, as well as the fibrin here. And well, this is kind of phenomenological modeling. So what we assume is that at the interface between the solid and the liquid, which we assume to be located at F, at the level Fs is equal to a half. So you should imagine that this is a three-dimensional level surface of Fs is equal to a half. There we assume that the cells prefer to adhere. Okay, and so when they are, so you could also imagine that if the fibrin density is very high, then you know it's like Tarzan. I always compare this to Tarzan. So I don't know whether you saw these movies of Tarzan, and all these monkeys and monkeys that are moving around in all these trees in the Amazon. If the density of branches, of twigs and branches is too large. They can't move. And if the density is too low, they can't move either. So they can't jump from one tree to the other one. The same holds for Tarzan, he couldn't do that either. So he needed these things to move from tree to another tree and from branches to another branch. So the cells are like that. So cells are very human in that sense. And so we assume that there is no movement if the solids fraction is equal to 1, if the solids fraction is equal to 0. Yeah, okay, then you could have chemotaxis here, by cells that are swimming in the extracellular liquid. By the way, gravity, of course, it's also important here, but we kind of try to quantify that, but it was negligible indeed. And so I also wondered, so they did these experiments, in Amsterdam. I wondered whether what happened if you put the samples upside down, or just tilt them, but it doesn't really hardly any influence there. So gravity is, in that sense, not important. Okay, so we also incorporated probabilities for differentiation of, because it also turns out that in these studies that tip cells are able to differentiate to stalk cells, and stalk cells are able to differentiate to tip cells. Especially the part of differentiating tip cells to stalk cells, I was really surprised by that. I thought, I couldn't believe it. I thought, this is ridiculous. But it apparently takes place. So there is this study by Tamala and many other authors that actually showed that the tip cells could fall back in their performance of being stalk cells in their habits. So we use this kind of exponential probability distributions where the probabilities are also influenced by the chemistry that they experience. I'll leave the details out of it. So we have this kind of model description. So this is actually the description of the model. So I think it's sufficiently complicated in a certain sense. And so you can link all these things. We actually just wrote down the paper. So this is quite new work. So we did some finite element simulation. So we had this model for the cells that are moving around, by the use of these stochastic differential equations. But we also solved finite element. We also solved the problem by finite element methods in 3 dimensions with Euler backwards time integration schemes. Very standard, very simple. But we had some interesting issues there as well. And we used the Euler-Maruyama method for the stochastic differential equation, which is also a well-established Japanese method. Okay, so the weak form of the mechanics. So you multiply by test function, you integrate by parts. Well, you know it. And so especially, this is not so interesting. I think everybody knows it here. Only the more interesting term is, of course, how you treat the forces here.
So you haven't told us what sigma, the tensor you have.
The tensor?
Yeah.
Yeah.
How do you describe the mechanical properties of the fiber?
I'll come to that.
Okay.
I'll come to that later on. Yeah. Okay, so at this moment we just use simple linear elasticity. Okay, so we use simple elasticity, but indeed we want to go to direction dependency. And so this is something that we still have to do actually. Okay, so all these chemokines, Sue doesn't like this word, but all these chemokines, they satisfy this diffusion reaction type of equations. And like I mentioned earlier, the mesh is moving because of the mechanics, the cells are pulling, so you have movement of the mesh. And so you get this passive convection because of the movement of your domain. We have this kind of boundary condition. And we use 2 different approaches. We either use Green's fundamental solutions. Advantage of these solutions is that you are only required to calculate the solutions at specific points where you want to calculate the solutions. So that's nice. On the other hand, they can only be applied for very idealized circumstances. So we also use finite element simulations to tackle the real world. So we want to be realistic as well. And so I will predominantly speak about finite elements simulations later on. Well, you know this figure, right? This guy. So we... Sorry?
Also squashed.
Also squashed. Also squashed, yeah. Yeah, maybe. Yeah, Maybe it's a bit my fault, but because you know I put many pictures everywhere, but you know what it is. You know, I'm always constrained by space. You know, all these things, and probably the Mac also does something crazy to all these pictures. But I think this is a picture of a couple of years ago, and you didn't change that much. I think I see. But I think you also used to have a beard at a certain period, right? But yeah, that is gone as well. So you're losing your wild hairs in that sense. So we have this secretion of these chemokines or these cells of specific positions. We use Anotita's Matsumoto's approach, which he developed in 2012. I think you developed it for the viscoelastic cells. And so that's actually the approach that we were using here to describe your actins, I think, or some chemicals or whatever. So we have this weak formulation once again we have this weak formulation for this reaction diffusion equation as well as the so we use the material derivative here, and then we plug it in, we get this formulation, and then we use the transport property of the basis functions, which was originally proved by these guys, Duke and Elliott, I think. I also remember that you mentioned this during one of your talks, that you proved it yourself as well. Indeed, I did it as well. So we get this relationship. It makes perfect sense. Further, we also had for this total derivative. So you are moving in an Eulerian, in a Lagrangian framework. I always confuse these two words for some reason. Then you get this equation after some manipulation with the product rule as well. Then you get the following, so you can write this into this equation. Then, maybe I'm a bit too technical, I realize now. And so hence we have this, we end up with this equation. Then we can also use the divergence theorem and integration by parts on the diffusion term, especially on these two terms. Then we get this, and from that, okay, we use Reynolds. Was he German? No, he was English, right? Reynolds, so it's just Reynolds. It's Reynolds, yeah, it's not German, of course. And I'm confused with Leibniz. Leibniz, of course, was a German. And actually, actually, this is Leibniz theorem, but then for more dimensions, so that's why I'm confused with the Germans. And I should also confess, I have a German background as well. Then we get, so we use Reynolds transport theorem, we apply it to the equations. Finally, we get this weak formulation. This is very simple because now, okay, so this is the total derivative. And so incorporating as well the movement of all these, so you should imagine that all these elements are actually moving. And these elements are moving because of the contractions that the cells are exerting. If I'm running out of time, please tell me. But I think I'm still okay. I'm still okay, right? And so this gives a very simple equation. And once we discretize this, then we apply this finite element approximation as well as the Euler backward time integration scheme. So we end up with this equation. And then something funny happened. Something funny happened here because what we did, Franz, I think, maybe I'll show his picture later on. I think, I'll show his picture later on. Franz, my student, used these three-dimensional elements, and then at a certain moment, his diffusion coefficients were very small, and so he experienced some oscillations, some spurious oscillations. Yeah, of course, we were not so happy with that, because we use Euler backwards, so that is a stable time integration. And we also observed that these oscillations would actually die away in the course of time, which is also which reflects the stability property. So that's perfectly okay. But we had some spurious oscillations, and we still had in condition of stability, but we had these oscillations. And then actually, what we actually realized was the following. And that, maybe I should use another blackboard. Maybe some of you already know it. So that maybe some of you already know this. And I also, of course, when I thought of this, afterwards, I thought, well, of course, this makes perfect sense. How could I be so stupid, But sometimes you have to collide you have to bump your head sometimes. And what happened was actually, so we have this equation, right? So we have this, let's say, tau plus 1, okay, this m, tau, c tau, and let's say plus s times delta t, tau plus 1, times C tau plus 1. Okay, so imagine that we have this equation. We have to solve this equation. Okay, and so this is after discretization. So we get this system. So what we want to do is, of course, we want to solve this for this quantity. So we move this to the other side. So we get m. Okay, I forgot about the taus, if you don't mind. Okay, if you do mind, I forget it anyway. Times s times c. Okay, here I write it down. Is equal to m tau times c tau. So you get this equation. Yeah, and actually what happened was that in the finite element approximation that we used, you have this mass matrix, okay, and the elements of the mass matrix, Mij, are given by the integrals over your domain of computation of phi I times phi j, right? I think that everybody agrees on this, the omega. And what we also had was that these entries, all of these entries, so these phi functions are all non-negative. So that means that these guys, if you use linear elements, these guys are non-negative. But these are non-negative. And what also happened, of course, is that if you don't lump the matrix, and that's and that's exactly what we didn't do in the beginning. If you didn't lump the matrix, yeah, what happened then, of course, is that some of these entries were positive. Also when I was not equal to j. So that happens sometimes. And then, of course, if you have this matrix here, m minus delta t times s, okay, it's no longer an M matrix, if you know what I'm talking about. Maybe some of you know what I'm talking about. It's no longer an M matrix. And then, of course, you cannot expect anymore that you have monotonicity of the solution. So you violate all the maximum principles and these kind of things. And this is normally something that I teach the students. And so we got these oscillations indeed by that. And there is, of course, a way to suppress them. You can just simply lump the matrix. So that's actually what we did. So simply lumping of the matrix. So then you suppress this. But this is something that you will not experience, for instance, if you apply the finite volume method, or if you apply finite differences, then you will never see this. And so this is really something, a drawback in this sense for the finite element method. So if you lump the matrix, then you don't have this problem. But if you don't, then you have to satisfy this criterion. So your delta t must be larger than a certain.
Could you prove that mass lumping was
yeah.
give you stability?
Yeah,
could you
yeah, that could we could prove that, because then that's very simple, because what you get is that this matrix m minus delta t times s, so the off diagonals are only determined by s, okay, and this s satisfies the right structure, so there's an M matrix, so you get an M matrix then, and the M only makes it better, so you get an M matrix, so you can easily prove that. So yeah, afterwards, I think, yeah, okay, this is very trivial, right? But on the other hand, we just experience that first. So sometimes you have to experience it. Then, your Gaussian orientation, indeed, very good. That's one of the questions, indeed. So the next thing we are interested in is indeed, and this is actually in the framework of the birds, okay, especially in the birds. Well, what happens is, maybe I can show the picture here. If you look at healthy tissue, this is a rearrangement of the extracellular matrix. In healthy tissue, what you see is that the orientation of the fibers is kind of random. Okay, so it's kind of random. So you get this kind of isotropic behavior of the tissue. It's more or less random. That's what the doctors show me. Okay, doctors show me this type of micrographs. Whereas, if you had a burn and the fibroblasts are healing, or are actually producing extracellular matrix, they seem to produce extracellular matrix in the direction of their motion. So what you get, if they are moving in the same plane or in the same direction, you get a very well oriented piece of collagen. I was so happy to be here in Cambridge, because what I saw was, for instance, and I got many ideas from that. I also saw that in tumors, in tumor islets, for instance. It seems that if you have these islets, they are surrounded by fibrous tissue, or they are surrounded by extracellular matrix that is actually oriented parallel to the perimeter or to the circumference of these tumor islets in such a way that the T cells, the T lymphocytes, are not able to get into these tumor islets, and so that they are not, and thereby they will not be able to kill or to battle the tumor cells. And so I have some ideas about that, but maybe I'll say something about it later on. But that's not what I wanted to talk about now. But in skin you also see that. So in burns, in newly generated extracellular matrix after burns, you will also see that the alignment of the collagen is oriented towards, the paths of motion of these fibroblasts. So you get a very non-isotropic structure there. And what we try to do here is we use the model by Cumming. So I didn't develop this myself. This model was developed down under in Australia by Cumming and his co-workers, and they introduced a symmetric orientation tensor. So they introduced this tensor, and what you can do is you can use this tensor. You can compute the trace, for instance. If you compute the trace, which is just the sum of the diagonals, then you get the collagen density. And you can also, if you calculate the eigenvalues and the eigenvectors of this tensor, then you also get the direction of the alignments. So if, for instance, if you have a zero eigenvalue in one direction, and you have, for instance, an eigenvalue is equal to 1 in the other direction, okay, so you normalize the eigenvectors, of course, which are, of course, because of the symmetry, they are orthogonal, they are perpendicular to one another. And you see that you have a very preferred orientation towards, let's say, the x direction. Okay? So this is what we are using now for the description of the anisotropies. And I have to confess, indeed, coming back to your question, indeed, with respect to the elasticity modulus, I think this is very important for the elasticity modulus. This is still something we have to work on. We didn't do that yet. So we only use this tensor for modeling the contact guidance of the fibroblasts. So they prefer to move, and we assume that they prefer to move according to the alignment of these collagen fibers, and which makes sense because it was so funny to see. Well, actually, it's not funny, but it was so interesting to see 2 weeks ago in one of these talks that I saw at the Downing site. We were the Downing site. That all these T cells were kind of moving around these tumor islets in kind of circles. That's a very frustrating thing to see, in fact. So we try to adopt the orientation of the tissue according to the movement of the, so this is something that we included in the modeling, according to the migration path of the fibroblast. So G here denotes the unit vector in the direction of the velocity of a fibroblast. So that's a unit vector. So we incorporated this equation, and this is the indicator function which actually denotes the space that is occupied by the fibroblast. You can also demonstrate very easily that if you compute the collagen density from this, which is the trace of this quantity, so then you only use the diagonals of these matrices, then you can also easily demonstrate that actually that this is a stable solution. And it's very easy to demonstrate this. It's very easy. If you want, I can do that on the blackboard. It's very easy. Further, so we have this cell migration also in the context of burns. So we have this Wiener process, random walk. We have the velocity of the cells by the deterministic, by the mechanical signals. You could also imagine that if you have a cell, for instance, if you have a cell that is walking, let's say, in an extracellular matrix and it's exerting forces, right? So the extracellular matrix is deforming. So you have displacement, you have a displacement field. And of course, if you deform this, well, maybe I shouldn't do this because this belongs to the university, but if I use my, so there's nothing in there, right? So if for instance these cells are if these cells are moving around here, and if I start pulling here, then of course this will all deform. So they will also have some component for the velocity by just mechanical drag, right? So where's my pointer? Oh, here it is. So that's something we also try to incorporate, the contact forces again, by army. And here we have the chemotaxis. This is the movement according to the concentration gradient of a certain chemical, as well as the contact guidance. So this is our tensor again. Maybe I should go to the fun part now, to the simulations. Okay, so like I mentioned earlier, if it comes to angiogenesis, right? My colleagues in Amsterdam measured the cavities. So they looked at the cavities that they had on top of the fibrin. Remember that? I said it in the beginning of the talk. So we are going to do the same here, because we want to match our calculations to their measurements, right? But what they actually did was the microscope was a bit had a bit of a kind of a funny focusing behavior. So here you can see these black patches, which actually represent the boundaries of these cavities. So they actually computed in Amsterdam. They computed the area of all these dark patches here. So of all these boundary segments. So that's what they did. Yeah, I didn't know that, but I would have preferred that they computed, let's say, these areas here. But that's not what they did. So we also had to do that in some extent. So let me show the simulation. So here you can see, before I run it, so here you can see the fibrin. This is the fibrin, this represents the fibrin. Here we have a colony of cells that are residing on top of the fibrin. Okay, and this is the what I showed earlier. This is just this top layer of the fibrin, and here we see some projections, you know, from different angles. And if you look at this movie, and of course you can see the following. So here you can see this at the top of the fibrin. So here you can actually see these cells. These cells that are have different colors and are located on different depths because of the focal properties of the microscope. They could actually see a layer of cells, and so we have cells that are on top and on the bottom and in the middle. And here you can actually see the development of these fibers and of these cavities. Furthermore, if you look at this three-dimensional picture, here we sketch the level curve of Fs is equal to a half, which kind of is taken as representative for the boundary, for the interface between the extracellular matrix, extracellular fluid, and the other substances, the other chemicals, the other phases. And these are the cells. Oh, it's not so easy to see. This is Frans Buchholz, by the way. This is the student who did it. This is my MSc student who actually did this. And so he developed this finite element code. He implemented the whole model, in fact. So this is quite a nice piece of work in 9 months, I think, what he did. And this is where he ended, okay? And what you can see here is that in this sketch, you can see that there's already initiation of branching of these things. And in some other movies, he made many movies. I think he made, maybe hundreds of movies. You know, he made hundreds of movies, and so we also collected the data. We also assembled we also accessed all the data. Did some statistics on that. I'll show that a bit later on. It's a lot of work. He did a lot of work. He really did a lot of work, and so he was also rewarded with a very good grade. Of course, there's no denying. And what we could also see is that there's this kind of branching, as well as that sometimes we could also observe that these branches were merging. Okay, so we also had this merging. So this is really nice here. Yeah, of course, these models contain stochastic processes like the migration of cells, like the differentiation of cells, and all these cellular processes. So you have to do, I don't know whether you have good eyes, you have to do some statistics. So we also sketched some, so we did many simulations. So for each run and for each case, we performed about 12 simulations in 3 dimensions. And then we did some statistics. We computed the mean, you know, the averages, and of course the standard deviation, confidence intervals, and this kind of thing. So you can see the confidence intervals if you look very carefully. So we also looked at the percentage of sprouting area. We looked at the perimeter, like I sketched earlier, like I said earlier, the perimeter, as well as the percentage. And so we looked at, let's say, something like the volume percentage of all these sprouts within the fiber network. Well, I think maybe I should skip this because I want to show you some more results that are maybe a bit nicer with respect to movies, etc. So I'll skip these results, but we looked at the influence of the vascular endothelial growth factor, so the impact of different things, of different parameters on the simulations. And because some parameters were kind of, well, known, okay, between quotes from the literature, and no between quotes, because it's always hard to get these parameters. And it's, for the case that you're asking this, is a very nasty question always. How do you get the parameters? It's very hard. We get them from literature as much as possible. We speak with our colleagues at Amsterdam about them. But in some cases, we just have to use educated guesses, right? We just use an educated guess for the parameters. Okay, that's all we can do in some cases. And so therefore we had to change, we had to vary these things, these parameters quite often. Well, the people in Amsterdam provided us with the experimental data for the perimeter of these cavities. So that's what you can see here. So they did experiments with dermal tissue endothelial cells, and also, like Amit is interested in adipose tissue endothelial cells. And so they used different types of chemicals to stimulate, for instance, the vascular endothelial growth factor, as well as the tumor necrosis factor. We did the simulations here. So these are the experiments, these are the simulations, and you can see that there is a discrepancy. If you look at these simulations, you can see that there is. So that the correspondence is not perfect. It's actually far from perfect, I think. It's far from perfect. So to be honest, it's very, actually I think this indicates that still a lot of work needs to be done. So we are not there yet. But I think, but what we can show from this is actually that, and that is actually that was my aim, and that was answering the question whether or not we could use this type of cell-based modeling to simulate a process like angiogenesis, vascularization. I think the answer is yes, we can. I think the answer is yes. But the answer is also, but. There's also a but. But there needs to be more work done. Because what you see here is that the sprouting assays will predict a monotonous behavior, monotonous increase with the concentration. Okay, but this is also what you would expect. And of course, if you look at if I look back at the figures that I showed earlier, if you look at the, let me see, so this is the perimeter, so what, okay, this is the probabilities. So here we also played with the vascular endothelial growth factor. And if you look at the volume, indeed, the higher the concentrations, the higher the gradients become. So the more chemotaxis you have, so the faster vascularization. That's also what we see here. If you look at the volumes, then it's okay, right? However, if you look at the perimeters, then we see that there is some discrepancy here, that we see that there is a decline at a certain moment. Well, that is something that I didn't like, I didn't like this. So when I saw these results, I think, well, this is not right, this is bad. But then I realized later on that if you look at the chemotaxis, and if you would have, let's say, because the main signal for the vascularization in this model is chemistry. Okay, in this model is chemistry. Okay, whether or not it's really biologically sound, because the mechanics is also important. That's another discussion. And what you can see is that if the concentration gradients are higher, then the amount of chemistry to the migration cells is larger. So the proportion of, let's say, the mechanical repulsing factors from the cells that are pushing away each other, as well as random walk, is also lower. And that is, I think, the reason why that the model predicts at a certain moment a decrease here because the cells are going directly, they are going down under directly. They penetrate the fibrin directly. And then Sue Gibbs also saw these results, and she said, yeah, that's right indeed, because so they also did some measurements, but I didn't use the parameters for those, or my student didn't do that yet. And that's something we should do, I think. they also did the same experiment. So this experiment was done with vascular endothelial growth factor, but they did the same experiment with tumor necrosis factor. And then they saw some similar behavior here. So I think that this could be right in a certain sense. But maybe we have to change parameters and this kind of thing. Okay, so then I'm going back to the burns. Okay, do I still have some?
5 more minutes.
5 minutes? Okay, 5 minutes, okay. So I go back to the burns, because I think this is a very important project for me, because I get a lot of money from the Burns Foundation, so I should say something about it. And of course I thank the Dutch Burns Foundation for this. Let me think that immediately now. And so what we see here is a schematic of the surface of the skin, where there is, let's say, a burn here. Of course, this is just a cartoon of reality. I realize it fully realize it. The pluses here are the orientation. Denote the orientation. So in this case, there's equal orientation in the horizontal and the vertical direction. So that means we have an isotropic medium here. As well as these blue things here, these blue circles represent the positions of fibroblasts, okay, which are responsible for the generation of extracellular tissue in the skin. And what they will do is, yeah, maybe I shouldn't say it, but show it, is that they will move into the wound, right? According to some chemical signals, and here we also took into account all the mechanics, so you can see that the wound is contracting here, and this is something that the doctors really like very much to see. So now here I stop the simulation, but you can see here that there is a preferred orientation of the collagen. Okay, so we also model the orientation of the collagen. We solve the mechanics equations. However, we do not yet use the collagen orientation as an input into the mechanical model. That is something that we are planning to do. So we are actually working on that, but we didn't yet have any simulations. And so here you can play with the wound area. So you can play with so initially it was normalized to 1, let's say. In the beginning, you will have these fibroblasts that are pulling already on their substrate, on their environment. So the wound is actually kind of expanding a little bit here. Whereas later on the fibroblasts get into the wound, then the wound will be contracted. This is not the healing phase, but this is contraction. Okay? And then, after a while, the myofibroblasts, okay, so the myofibroblasts will die by apoptosis. And then there will still be residual stresses. By what I showed earlier. I'm not going to show it again, right? Because otherwise, and so that's actually what you see here. So this is the case of a healthy wound where everything goes back to normal, let's say, and then of course, the top layer can be healed by let's say, the cell closure, the wound closure. And so we can play with many of the parameters here. And of course, we also should realize that there's stochastical signals, so we should take that into account. I also had a cartoon model with Amit. So last year we met in Sweden, I think, on a conference. And we also we developed a formalism for cell differentiation of mesenchymal stem cells. They could either differentiate to adipose cells or fat cells or to muscle cells. So the red ones here, so these are all stem cells, in fact. And what we assume in this model, if you look at many of cell differentiation models, in particular if you look at the models that are based on, let's say, partial differential equations, so most of the time what you see is that they treat cell differentiation as kind of a reaction term. So you have a differential equation and then there's a reaction term, and so from one cell phenotype it's a loss term, and to the other to the other phenotype where the cells are differentiating to, it will be a source term. So positive, negative. So you get something like this. So what you see is that cells will actually start differentiating immediately. However, some biologists also told me that cells merely gradually adhere properties of the phenotype they are differentiating to. So what we did in one of the PhD studies of one of my students, Pavel Progaro, we developed a continuum scale model, so partial differential equations based model, which I'm not going to show here, and in which we introduced a maturation space. So we had differential equations, let's say, with 2 or 3 spatial dimensions, as well as some other dimensions for the differentiation space. So we get a 5 or 6 dimensional differential equation. Of course, these things are very expensive to solve if you have many phenotypes, so it becomes very expensive. And what we also assumed in the model is that cells could adhere either of the phenotypes they are differentiating to. And so you can see it like this. And so if you consider, let's say, this differentiation space, okay, imagine that here I have a stem cell. So this is the stem cell. Okay, this is the stem cell, mesenchymal stem cell. And they could differentiate, let's say, to cell type adipocytes, or they could differentiate to muscle cells, or for instance to bone cells, whatever. You know? And what we assumed in all of these models, that cells can gradually adhere the properties of, let's say here, if this is equal to 1, let's say, and here we have 1, you know, so 1, 0, 0, 1, 0, 1, 0, 0, 0, 1. So here there are fully adipocytes, here there are fully bone cells, osteoblasts, and here there are fully myocytes or muscle cells. So what we assume is that the cells, they can kind of gradually move towards, and here in this path, they would actually gradually acquire the properties of fat cells. And this is actually what Amit also says, that models with this experiment, that you can see that there is a gradual development of lipid droplets. So here, let's say the cell starts developing these lipid droplets, and these lipid droplets will grow and grow more and more and more. So they will push on their environment, on their membranes, and in such a way that at a certain point they start losing their mobility. Okay, they start losing their mobility. So they are there, you know, just fat people, like fat people sitting on the couch, indeed. So they are there. Whereas here we have the same for the muscle cells. So they start exerting, so they become stronger. They are like bodybuilders. So here are, let's say, the fat people, and here we have the bodybuilders. Bodybuilders. Yeah. I have a slide to dyslexia. So here we have the bodybuilders as well as the and this is actually, so we developed this into a partial differential equations formalism. Okay, if you want, I can give you the references. But we also lately incorporated these things into the cell-based model. So what you can see here are, let's say, mesenchymal stem cells, where the bluish cells represent cells that are acquiring the properties of fat cells. So they are pre-adipocytes, I think. They are kind of pre-adipocytes. Whereas here, the red cells are either muscle cells or are pre-myocytes. So they are adipocytes or pre-adipocytes. So, well, then what we did in this model, we also we use this formalism, okay, for the stresses and for the force that the cells will exert. And so we also assumed that there was some cyclic loading, and according to some cyclic loading, the cells would either differentiate to fat cells or to muscle cells. Furthermore, what we also, what else do I need to say? Is there anything else? Yeah, we also assumed that cells, they are moving throughout this, let's say, this maturation space, and that they can only move over the axis. Okay, they can only move over the axis here. Sur les axes, over the axis. They can only
go down.
Sorry?
They can go down.
Yes, they can go down as well. So, for instance, what could happen is that indeed the pre-adipocyte, I don't know whether this is biologically sound, but that's in the model at least, that a pre-adipocyte would, for instance, differentiate more towards adipocyte, and then if it receives signals, I know what I wanted to say, and if it receives signals from periodic cyclic loading, in that case it could go back. Okay, then the cell wants to go back to, let's say, the stem cell, and then it can go back to, can differentiate towards a muscle cell. But it's not something like this.
And they cannot do up, because I don't understand what is a cell doing both. What is that?
Oh, here, this one you mean?
Yeah,
Oh, this is just another phenotype that we added. So So these stem cells could differentiate, for instance, to many different types of cells, to many different phenotypes.
Something else.
This is just something else. So
Okay.
in this simulation, I'm actually only considering this. Okay, so that's what I'm considering here. But of course, you could extend. We extended the formalism to let's say n phenotypes. Yeah. And another thing I wanted to say is that apart from this mechanical signal that the cells are experiencing, and as well as they are exerting on their environment, the cells are also exerting chemicals. So these chemokines. So the fat cells, they will secrete a certain chemical, as well as the muscle cells, and by this chemical, all these cells, and by the mechanical strains as well, all these cells want to behave like their neighbors, right? So yeah, it could also be a human characteristic. We want to fit well in a certain group, although we also find some people that take the lead, like the tip cells, that want to be different. So there's also people that want to be different from the others. Oh yeah, okay. So let's have a look at that movie, right? Because I talk too much, don't I? I was meant to give a talk, so I talk. So okay, I just gotta stop here. So here you can see that the cells are moving around a bit, and at a certain moment they lose their ability as they get fully differentiated. And so this framework indeed was we just had this was also a master's project of one of my students. I didn't have his picture here, Sepp Harleveld. And Sepp Harleveld is a kind of student that doesn't want to behave like his neighbors. So that's a very but I like these people, you know. I like these people so because they come up with nice ideas and so on, you know. So that was and here you can actually see that there is a higher proportion of muscle cells. Now, one question you could ask, of course, if you look at the, of course, we can sketch many diagrams from this, you know. But one question you could ask is, does the model only predict two possible outcomes? Is the model only capable of predicting two different outcomes, in the sense that either my whole population will be muscle cells, will I get a population with only consisting of muscle cells in the end, or will I have a population with only fat cells? Well, I was afraid of that the model would predict it in this way, that we would only have, let's say, muscle cells or fat cells, you know, and then of course there should be else. Of course the model is not ready yet, you know, it's not finished yet. Models are never finished. And what we see indeed is that in the calculations is that the model will actually indeed predict that in the domain of computation there will be some patches, I mean regions, which are either muscle cell dominated or fat dominated. But you see patches, so you can see patches of clusters, or clusters if you like, of fat cells and of muscle cells. So the answer is no. So there is room of coexistence of these cells in the end. So that's, I think, very nice. I could give a separate presentation about this work, I think, but I'm not going to do that now. So I'm heading for the conclusions now, indeed, and that is, my main part of the talk was angiogenesis, like I mentioned earlier. So these are just small excursions that I mentioned here. We indeed have a cell-based semi-continuum model for angiogenesis. I think it's possible. So the answer was yes, we can. However, there's also however, we should do more work. So we have a nice qualitative morphological agreement, I think, with a model. The quantitative agreement is something we should work on. So, but that's always a good excuse. Oh, I have some more results indeed concerning the evolution of cell shapes. I added this slide because this is actually the work of one of my bachelor students in Delft in mathematics. So what he did was it was a very, very nice project also. So here you can see, let's say, an immune cell. And you can think of a T lymphocyte or something like that, or whatever. And here there's these two circles are barriers. And this thing here is, let's say, a bacterium. So this cell is going after this bacterium. And this bacterium is releasing, let's say, some, by this bacterium, by this pathogen, for instance, the result of that is, for instance, a concentration of a certain acidic chemical. You can think of a lactate or something like that, a biotic lactate. And this, the concentration gradient of this biotic lactate makes this cell move towards that concentration according to chemotaxis. So let's have a look at what happens here. So here it starts. And the nucleus is also, that's also something new indeed, the nucleus is also deforming. That was an essential thing because our joint friend, so Amit Kevins and my joint friend Daphne Weiss, who Amit mentioned earlier, is actually doing research on cancer cells. And what she saw was that sometimes cancer cells would use, okay, oh sorry, yeah, should I stop?
I think so.
Yeah, okay, I stop. Okay, yeah, okay, I stop there. Let me show the last movie that I think that I should pay some tribute to my student here. So this is the three-dimensional version here. You can actually see how the cell captures this thing, and also the nucleus inside. You can't see it, of course, but the nucleus inside is also changing shape, as you can see here. So here we plotted the nucleus as well. So some more cells that are coming together. This is a bachelor study of about 3 or 4 months of one of my students. I'm very grateful to be here. Thank you.
One quick question.
Maybe I talk too much, maybe people are hungry and want to go for lunch.
Everyone-
I'm around you for 2 more days.